The human brain is supposed to liquefy within days of death. Ask any forensic scientist, any undertaker, any embalmer, they'll tell you the same thing. The brain goes first. It turns to soup while the bones are still intact. For more than a century, that assumption has shaped how archaeologists understood soft tissue preservation. They looked for mummified skin, for frozen muscle, for the tanned leather of bog bodies. Almost nobody looked for brains. Then a former undertaker named Alexandra Morton Hayward started digging through 400 years of archaeological literature. What she found upended everything we thought we knew. The textbooks were clear on this point. When a human body decays naturally, without embalming, without refrigeration, the internal organs break down in a predictable sequence. Bacteria from the gut spread through the bloodstream, consuming tissue from the inside out. The brain, soft and rich with water, succumbs quickly. Within 48 hours in warm conditions, it begins to liquefy. Within a week, only fragments remain. This understanding drove archaeological practice for generations. When researchers uncovered ancient human remains, they cataloged bones and teeth. They looked for preserved skin in desert burials, for tanned tissue in peat bogs. They did not open skulls expecting to find anything intact. Why would they? The brain was supposed to be gone. Yet scattered through centuries of excavation reports, something kept appearing that didn't fit the model. A shrunken mass inside a skull, a fragment of tissue with recognizable folds, notes scribbled by archaeologists who weren't sure what they were looking at. Each report called its find unique. Each described it as exceptionally rare. Nobody connected the dots. As early as 1904, the Egyptologist Elliot Smith had complained about the scanty literature on preserved brains. He argued that almost every archaeologist who excavated burial sites was aware that brain tissue appeared in a large proportion of crania. His observations went largely ignored. The field had already decided that brain preservation was anomalous, and anomalies don't demand explanation. 3 factors locked this assumption in place. First, the labeling problem. When excavation reports described preserved brains as unique or one of a kind, they inadvertently restricted access. If a specimen is truly irreplaceable, you don't risk destroying it with invasive analysis. Less than 1% of all documented preserved brains have ever been studied for ancient biomolecules. Second, the expectation gap. Researchers expected to find preserved brains only under extreme conditions, frozen in permafrost, dried in desert sand, tanned in acidic bogs. They didn't expect to find them in wet environments, where water should accelerate decay. When brains appeared in lake beds and shipwrecks, the finds seemed impossible, so they were treated as curiosities rather than data. Third, the fragmentation of knowledge. Reports of preserved brains were scattered across journals in dozens of languages. An excavation in Egypt might be published in French. A bog find in Denmark might appear in German. Nobody had ever tried to gather them all in one place. Morton Hayward, working at the University of Oxford, had spent years as an undertaker before turning to science. She knew decomposition intimately. She had seen bodies liquefy in summer heat and freeze solid in winter morgues. So when she came across report after report of preserved brains, she started asking a question that should have been asked decades earlier. How rare could they be if they kept turning up? The answer would take years to assemble. Morton Hayward and her team combed through excavation records in more than 10 languages, tracking every mention of preserved brain tissue in the archaeological record. They contacted researchers on 6 continents. They cataloged specimens from desert tombs and sunken shipwrecks, from lead coffins and mass graves, from shallow pits and sacred pools. When the database was complete, it exceeded all previous compilations by a factor of 20. The final tally was staggering. More than 4,400 preserved human brains, documented from 213 unique sites across every continent except Antarctica. Not across millions of years of human history, across just the last 12,000. The number alone shattered assumptions. But the details were stranger still. When the team sorted the brains by preservation type, 4 mechanisms were familiar. Dehydration preserved some brains in scorching deserts. More than 500 specimens from a single predynastic cemetery in Upper Egypt dated to roughly 6,000 years ago. Freezing kept others intact in mountain passes and polar regions. Tanning occurred when peat bog chemistry transformed brain tissue into something resembling leather. Saponification turned fats into a waxy substance called adipocere, what forensic scientists grimly call grave wax. These four processes were well documented. They explained why brains sometimes survived alongside other soft tissues. But almost a third of the archive, more than 1,300 brains, didn't fit any known mechanism. In these cases, the brain was the only soft tissue preserved. No skin, no muscle, no organs, just a shrunken mass rattling inside an otherwise empty skull. And these brains were older than any of the others. Some dated back 12,000 years, to the end of the last ice age. In a laboratory at Oxford, Morton Hayward now houses more than 570 of these mysterious specimens. 4.5 refrigerators filled with ancient brains that should not exist. The breakthrough came when the team mapped each brain to its burial environment. Known preservation methods followed predictable patterns. Frozen brains came from cold regions. Dehydrated brains came from deserts. Tanned brains came from acidic bogs. But the brains of the unknown type, the ones that persisted for millennia with no other soft tissue, showed a different distribution entirely. Most came from wet environments. Lake beds, the bottoms of wells, shipwrecks, places where water should have accelerated decay. Consider the evidence from Windover, Florida. In 1982, a construction crew draining a pond for a housing development uncovered human skulls. Archaeologists eventually excavated 168 skeletons from the peat, a Stone Age cemetery submerged in a small lake for 8,000 years. When they opened the skulls, they found brain tissue in 91 of them. Some brains were intact enough to reveal individual structures. Cell architecture was visible under a microscope, and scientists extracted DNA, the first time ancient genetic material had ever been recovered from brain tissue. The Windover brains should not have survived. Florida's climate is hot and humid. Organic material rots quickly. Yet something about the pond's chemistry, the peat, the lack of oxygen, the rapid burial of bodies within 48 hours of death, created conditions that preserved neural tissue for 80 centuries. Or consider Heslington, near York, England. In 2008, archaeologists excavating land for a university campus found a skull buried face down in a waterlogged pit. When a conservator noticed yellowish material inside, they sent it for imaging. The scans revealed a human brain, shrunk to perhaps 20% of its original size but with gyri and folds still recognizable. The texture, one researcher noted, resembled firm tofu. The skull dated to the Iron Age, roughly 2,600 years ago. The man had been struck on the head, then decapitated with a thin blade and buried immediately. No other soft tissue survived. But when scientists analyzed the brain, they identified more than 800 intact proteins. Some were in good enough condition to trigger an immune response in modern laboratory tests. The proteins had folded into tight, stable aggregates. This folding, researchers believe, may hold part of the answer. In living brains, proteins maintain flexible structures. After death, enzymes should break them down. But in the Heslington brain, something deactivated those enzymes within months of death, possibly an acidic fluid that seeped into the skull cavity. The proteins locked into place, and they stayed locked for 26 centuries. What this shows is unmistakable. The brain is not simply soft tissue waiting to decay. It has a unique biochemistry that sets it apart from every other organ in the human body. Neural tissue contains an almost equal ratio of proteins to fats, roughly 1:1, with very little carbohydrate. Most other organs are loaded with sugars. The brain's transmembrane proteins are unusually rich in sulfur-containing amino acids, particularly cysteine and methionine. Under certain conditions, these molecules may cross-link with each other, forming stable polymers that resist bacterial attack. Add trace metals like iron or copper from surrounding sediment, and the tissue may undergo a kind of natural fixation. The metals bind to protein structures, locking them in place. In time, the organic material may even begin to mineralize, becoming in effect a fossil of its former self. This rules out coincidence. Brains don't persist by accident in wells and shipwrecks and lakebeds. The pattern is too consistent. The chemistry is too specific. Something about neural tissue makes it uniquely capable of long-term survival under conditions that destroy everything else. And this implies something remarkable about what we've been missing. For more than a century, archaeologists have been discarding the most information-rich organ in the human body. The brain is the seat of memory, of personality, of the biological record that defines an individual life. Soft tissue preserves ancient DNA far better than bone. It preserves proteins that reveal diet, disease, and metabolic history. It preserves lipids that can be analyzed for evidence of neurological conditions. Fewer than 1% of the brains in the archive have ever been analyzed for these biomolecules. The untapped potential is almost impossible to overstate. Not everyone agrees on what the findings mean. Some researchers urge caution, noting that the unknown preservation mechanism remains uncharacterized. The proposed explanations, molecular cross-linking, metal complexation, mineral precipitation, are plausible but unproven. Without controlled experiments replicating ancient burial conditions, the theory remains a hypothesis. Others raise methodological concerns. The archive relies on published reports, many of them decades old. Standards of documentation varied. Some descriptions are vague. A few specimens may have been misidentified. The true number of preserved brains could be higher or lower than the current count. Morton Hayward's ongoing research at Oxford aims to address these gaps. Her laboratory now contains more than 570 brains of the unknown preservation type. Using mass spectrometry and other analytical techniques, her team is working to identify the precise chemical signatures that distinguish long-lasting neural tissue from tissue that decays normally. The ethical questions are unresolved. Many cultures regard human remains as sacred. Some indigenous communities have fought to repatriate ancestral bones from museum collections. The decision to extract and analyze brain tissue, even tissue that has lain in the ground for millennia, raises questions that the field has only begun to address. What remains undisputed is the scale of the discovery. 4,400 brain, 213 sites, 6 continents. A 12,000-year record hiding in plain sight because no one thought to compile it. In Sweden, 8,000 years ago, someone mounted a human skull on a wooden stake and placed it on a stone platform at the bottom of a lake. The victim had been struck on the head, hard enough to fracture bone, but the wound had healed. Years later, for reasons we cannot know, this person was decapitated and displayed. When archaeologists excavated the site in 2012, they found brain tissue still pressed into the interior of the skull. That fragment of tissue, preserved since the Stone Age, carries information we are only beginning to read. What diseases afflicted this person? What did they eat in the final years of life? Where did their ancestors come from, and where did their descendants go? The molecules that might answer these questions have survived 8,000 years underwater. We assumed the brain was fragile. We assumed it vanished first. We assumed that finding one intact was a miracle. But the evidence now points in a different direction. The brain may be the most durable soft organ in the human body, under conditions no one thought to examine. 12,000 years of human history are preserved in specimen jars and refrigerated drawers across the world. They've been there all along. We just didn't realize we should be looking. If the brain can survive when everything else decays, what else have we overlooked?